Hello, this video is going to cover our phases of the cell cycle, mitosis, and some cyclin and CDK activity. So we're going to start with just our overall cell cycle. Okay. Actually, let me use this color. So at the top, use one color. That's not a very even circle, but you get the point. There's another bad circle. Okay, so this top, actually we're gonna start on this arrow. So this arrow here is our G1. So that's when we're gonna replicate RNA, proteins, and organelles. Okay. And then this is G0. This is our specialized resting state that the cell can go in. It's still an active functioning cell. It's just not going through the rest of the cell cycle for the time being. So this little extra circle here is G0, so that's our resting state. Sorry, my cat's being loud. It's our resting state. Resting state. And then if the cell comes into the resting state, it can either go there temporarily or permanently. So if it wants to go temporarily, that's called quiescence. Quiescence. And then, that's temporary. And then senescence is permanent. Senescence, permanent. Okay, and then this next phase is our S phase. Okay, and that's when we're gonna duplicate our DNA, and that's it, pretty much. So our DNA duplication or replication, or synthesis. Right, so if you think synthesis goes with the S phase, that might help you remember. Okay, and just as a reminder, we have 46 chromosomes in our normal body cells, so we're replicating all of those. And then our last blue arrow is our G2 phase. And this is pretty much just a checkpoint, just to make sure that everything was replicated well and that we're ready to go into the next part of the cell cycle. So it's pretty much just a checkpoint. To make sure that all is good to go. And that's all. Oh wait, and then, duh. Okay, so all of this blue is interphase. So I'm gonna write it in blue. And then all of the red up top, just the one arrow, is our M phase. So our M phase is composed of mitosis and cytokinesis. So we'll go over every step of that, but mitosis is when we are replicating all of our genetic, when we're dividing all of our genetic information because we replicated it here. And then cytokinesis is when we divide the rest of the cell, so the cytoplasm. So just to complete this look, this was the cell cycle. I'll label that. Okay, so we had our G1 starts out interphase, S phase, G2, M phase, and then we have our G0 that we could go off and rest in. And that's it. And then, so we're gonna go through all of the phases of mitosis. I'm gonna go ahead and erase this, so make sure you've got that down. There's a bunch of cat hair on it. Okay. So, let's see. Use this color. So our first phase is prophase. Let's see, 1, 2. Make sure you leave enough room on your paper because my board isn't big enough to. I might have to like erase stuff to make more room and not be able to finish all of mitosis on the same board, but we will try our best. So, can you see this? Yep. Okay, so prophase is our first phase of metaphase, of mitosis. Oh my goodness. Our first part of mitosis is prophase. So our cell, I'm gonna draw a little cell. We've got our chromosomes here. So like I said, we've got 46 chromosomes that all get duplicated, but I'm not going to draw 46 chromosomes over and over, so we're just going to put 2 of each. So like the blue got duplicated and the red got duplicated, just to represent that. And then let's do orange. So draw, these are called centromeres. Wait a second. Hold on, it'll come to me, but this is where the mitotic spindle comes from. So we're starting to form our mitotic spindle. Centrioles, sorry. Okay, these little orange nubbins here at each end, my bad. Okay, so these are called centrioles. That's where our mitotic spindle is growing out of. Okay, a little brain fart. So the big things that are happening in prophase is our chromatid has condensed into our chromosomes that we see as like little X's. So we condensed to chromatid and then condensed down to chromosomes. Hey, don't play with that. The cat was trying to play with the eraser. Okay, condensed to chromosomes. Our nucleolus is disappearing. So the nucleolus is kind of like the nucleus within the nucleus. It's what like holds the DNA together within the nucleus. So that is disappearing first. Whoa, nucleolus. Disappears, and then we're going to start forming mitotic spindle. Okay, so we've got our chromosome. Actually, I'm not going to draw it like that. Sorry. So this is just going to represent one of our little chromosomes that we've got in there. So in the middle of our chromosome, connecting them together, we've got our centromere and our kinetochore proteins, and those will come, they'll become important later. So the centromere is like the middle part, and then we've got some kinetochore proteins located there as well. So centromere and kinetochore proteins. Okay, good. So can we like box that off? Okay. And then next is prometaphase. So let's draw another little cell. So our nuclear envelope has completely disappeared now. Our microtubules are going to attach to the kinetochore. I'll draw that out in a second. Let's write it down first. So microtubules attach to the kinetochore. And then our polar microtubules are going to attach to each other. Okay. So our chromosomes still aren't perfectly centered, but they're on their way. So we'll draw them kind of like off center still. And then we had our centrioles out here, little nubbin looking things. And then I know I just drew like in a generic orange color in our prophase, but we're gonna color code our microtubules now. I need a different color. Okay. So our purple is going to be the kinetochore microtubules. So the kinetochore microtubules attach directly onto that kinetochore protein that we said was in the middle of the chromosomes. So the kinetochore microtubule is going to go to the chromosomes. And then we have, let's see if we can see this. Nope. What about this one? You see that? Not really. Can't we have a dark green? There we go. Okay, and then our polar microtubules are going to be dark green, and those are attached to each other. So just kind of have them overlapping in the middle. That's the polar microtubules. So polar microtubules was green. And then our kinetochore microtubules are purple. So what's going to happen is the kinetochore microtubules will pull on the chromosomes and shorten, and our polar microtubules will push against each other, but we won't get to that until anaphase, but just a little heads up. So, oh my goodness, dropping everything. Okay, so that was prometaphase. Let's do metaphase here because it doesn't have too much going on. So in metaphase, all that's going to happen is we line up our chromosomes on the metaphase plate. Everything else stays the same. So that is like a key word for you to remember is metaphase plate. That's pretty much all you need to know for this phase. So we're going to redraw our chromosomes, and they're going to be centered. So the metaphase plate isn't like an actual line that you would see if you looked at a microscope. It's just an imaginary line that the chromosomes line up on. Our little centrioles. And then we had our kinetochore microtubules attaching to the chromosomes. And then those polar microtubules attaching to each other. Okay, so this like center line, imaginary line that they're lined up on is our metaphase plate. Oh, that's not straight. That's okay. Okay, good stuff. So next is anaphase. So in anaphase, our polar microtubules will elongate and the kinetochore microtubules will shorten. So when this happens, we're going to separate the chromatids at that centromere, at that center part. So we're going to separate the chromosomes. At the centromere. Okay, and then we do all this with the help of motor proteins. Okay. So let's draw our little chromosomes being pulled apart. So each like half was its own copy of the chromosomes. So like originally it was a one line, and then it got duplicated to look like that X. So now we're splitting it up again, so that each new cell gets only one copy of each. Little centriole. So the kinetochore, we said, are shortening, right? So they are shortening to pull the chromosomes towards the end of the cells, towards the ends of the cell that the centriole is in. And then at the same time, the polar microtubules are getting longer and pushing on each other, and that will help push the ends of the cell away from each other and elongate the cell to even more separate those chromosomes. Calvin, that's not a toy. Okay. So that's anaphase. And then next is, telophase. So telophase is our last phase of mitosis. But remember, we still have cytokinesis after, which technically is not part of mitosis. So in telophase, our mitotic spindle is going to disassemble, so it goes away. Okay, and then we're going to reform the nuclear envelope because we're forming 2 different cells eventually. Whoa, okay, sorry. Reform nuclear, hey, Calvin, can you see his paw? No, envelope, okay. And I actually should have drawn this longer. Because remember, our cell was elongating with the help of those polar microtubules. And then we're also going to decondense those chromosomes back into chromatin. So at each end of the cell, we've got those chromosomes. Our microtubule, our mitotic spindle with those microtubules are gone. We're reforming a nuclear envelope around those. And then those will decondense back to be more like just a glob of DNA, but for our purposes, we'll continue to draw them like that just because it's easier to visualize. Okay. And then, so all of these, 1 through 5 is mitosis, and then cytokinesis is its own guy. So cytokinesis, think cytoplasm, right? We're doing cytoplasmic division. Hey, I'm doing a video here. So what's going to happen? I'm going to draw on the telophase one. So we are going to split down the middle here pretty much. Actually, that's exactly what we're doing. So we're going to form a contractile ring. So we could actually kind of make this look like a little ring. So kind of picture that going all the way around the cell. So we're forming a contractile ring with actin around the cell. And so if my hand is the cell, it's going to like pinch. So it's going to squeeze the middle and pinch it. And we're going to form a cleavage furrow. And then it keeps tightening and tightening and pinching. And eventually it just pinches them apart. And we form 2 cells. So we've got a contractile ring. Which is actin, forms the cleavage furrow. And then we're going to end up with 2 daughter cells. Which are diploid cells. Hey. Here, play with this one. So those diploid cells have 46 chromosomes, just like the one that we started out with. Okay, so our new cells have the nucleus in them with the same chromosomes from before that we just split up. So that is cytokinesis. So all of this is our M phase. We got mitosis and cytokinesis. And then something else that we could do if we are forming haploid cells to form germ cells, then we do meiosis. So meiosis is completely different from mitosis. Like, it uses the same steps, but we pretty much just do it twice. You don't really have to know too much detail, just like general idea of what meiosis is. So meiosis is going to do division twice. So we're going to end up with 4 daughter cells. Which are haploid. So they're going to have half the amount of chromosomes that that our diploid cells have. So they're going to have 23 chromosomes. Which is good, because if we're making egg and sperm, which are the germ cells. So if we have the egg and sperm that come together, then the 23 plus 23 equals our 46 that we have to have for our, any other type of cells in our body. If any of the division steps in meiosis goes wrong, then we end up with things like trisomy 21, Turner syndrome, when like maybe too many chromosomes goes to one cell, like with trisomy 21, or maybe not enough chromosomes goes to one cell. That would be Turner syndrome. So remember those. Okay, and that's when division goes wrong. Cool. There's that. Now we're going to go through the cell cycle and talk about all of our checkpoints. So make sure you've got this, and you can see all that. Okay. I'm going to go ahead and erase it. Okay, big picture that you definitely need to know is that cyclin concentration varies and CDK concentration does not. CDK stands for cyclin-dependent kinase, so it's dependent on the concentration of cyclins. So cyclin varies, CDK does not. So we said CDK stands for cyclin-dependent kinase. Okay, and then overall view, when we phosphorylate things, we're going to phosphorylate the serine and threonine amino acids, and that'll give us some sort of cellular change. So let's draw a little chart here for cyclins and CDKs. Can you stop? So our cyclin D goes with CDK 4 and 6. Cyclin A and E goes with CDK 2. And then cyclin B goes with CDK 1. Okay. And then just to have like our little cell cycle over here. So our G1 is the only one that its checkpoint has like a special name. So our G1 checkpoint can also be called the restriction point. Restriction point. And then the rest are just called checkpoints. So we've got our S checkpoint. So this is our G1 phase, S phase, G2, M phase. So G2 has a G2 checkpoint. And then we have an M checkpoint. And then G0 doesn't have a checkpoint, it's just G0. Oh, I wrote on my pants. Darn it. Okay. So that's that. I'll just leave that. Okay. So our G1. So with G1, we've got our RB and P53 tumor suppressor proteins, so we'll do each one separately. So we'll do G1 RB over there and G1 P53, and they're both tumor suppressor proteins. So tumor suppressor proteins would like stop the cell in the cell cycle so that we can fix any damage and make sure everything's good to go so that we don't produce tumors. That's the whole point of a tumor suppressor protein. So we have a cell. I'm so sorry, my drawing skills are subpar today. There's a little cell. So our RB is doing what a tumor suppressor protein does, and it has stopped the cell in our G1 phase so that we can like make sure everything's good to go and there's no damage or anything, because after G1 we're going to go into S phase and duplicate our DNA, and we don't want to duplicate the DNA if there's something wrong with the cell, because that would just lead to all sorts of problems. So our RB is going to bind to transcription factors E2F and DP12. So those are transcription factors that would usually let the cell go into S phase, but we're going to stop it and hold it in G1 phase for now. So RB binds the transcription factors. E2F and DP12. Okay. So what happens is our Cyclin-D and CDK4/6 are going to bind and become active. So Cyclin-D, CDK4/6. So that's binding and making an active complex. And remember, it's a cyclin-dependent kinase, so it's going to phosphorylate something. So it's going to come in and add a phosphate to RB. It's going to hyperphosphorylate the RB, and that causes the RB protein to stop suppressing the cell, so that the cell is able to go into S phase. And we can continue the cell cycle. So cyclins and so the cyclin-CDK complex helps the cell continue on in the cell cycle. So that one's done. And then our P53. So we have a cell, and there's some DNA damage in the cell. So something has hurt. That's supposed to be a double helix. That's kind of bad. But something's wrong with our DNA in the cell. There's some damage. So our P53 is going to activate P21. P21 is going to come in and check out the cell, and it's going to try and fix our DNA damage. If it cannot fix the DNA damage, then we do apoptosis and kill the cell. But if we can fix the DNA, then we fix it and we go into S phase. Our P21, because it's holding the cell in G1 phase, it's an example of the Cyclin-CDK inhibitors. Sorry, that was hard to get out. It's an example of our CKIs, so cyclin-dependent kinase inhibitor. So P21 is an example of a CKI. Okay, so that's that. So if we have an HPV infection, that's going to make proteins E6 and E7. Makes proteins E6 and E7. So those are going to go, and E6 is going to affect P53 so that we can't stop our cell and fix the DNA. So it's going to go and still have damaged DNA and go into the rest of the cell cycle. So we can't stop and fix DNA damage if the E6 binds P53. And then the E7 protein would go and affect RB so that we can't pause the cell in G1 and check before we go into S phase. So our E6 would come affect, I should have drawn E6 first, sorry. E6 is going to come affect P53, and then E7 affects RB, and both of these will lead to unregulated cell cycle progression and probably cancer. Or something not good. Okay, so that's our G1. In G2, we have our cell. Nice happy cell. And what would usually happen, so this is getting stuck in G2 for a checkpoint. So we have a CDK1, and our CDK cyclins like to let the cell go into the next phase. But in order to stop it in the G2 phase, we have to temporarily inhibit the CDK1. So we've got CDK1, and we are temporarily inhibiting it with CDK1 inhibitory phosphotyrosines is what they're called. CDK1 inhibitory phosphotyrosines. I'm going to abbreviate that. CDK1 inhibitory phosphotyrosines. So those, whoa, I don't know what happened there. Those are going to inhibit our CDK1. So if we want to let the cell go out of our G2 and continue on the cell cycle, we have to get rid of this inhibition. So our CDC25C that's a lowercase d, sorry, CDC25C is going to come in, and it's going to remove that inhibition, and then that allows our CDK1, because it's not being inhibited anymore, it allows it to bind to cyclin B. So we form the complete CDK1, CDK cyclin complex. Talking faster than I'm writing. CDK1. So we've got our CDK1 cyclin B. So now we have an active kinase because we've got our active complex. So that is activated now. And so that's going to go and allow the cell cycle to go into our M phase from G2. Cool. So if we don't want to let our cell go from G2 to M, so we want to turn off this process, we have some other tumor suppressor proteins. They're called ATM and ATR. So those are tumor suppressor proteins. So they're going to come in and inactivate CDC25C so that we don't do this and we don't go into M phase. So they're going to come in and inactivate that CDC25C. So now CD25C is not able to come in and knock out our CDK1 inhibitory phosphotyrosine. So we're going to have this inhibition of CDK1, and we're not going to be able to form an active Cyclin-CDK complex. So our cell's going to stay in our G2. Okay. So that's G2. And then we have one last checkpoint that we're going to talk about, and that is our M checkpoint. I realize we have our S checkpoint, but we don't really talk about anything specific that goes on in that. Just know that there is a checkpoint in our S phase, but he gives you examples of G1, G2, and M. So our M checkpoint is in metaphase, right before we go into anaphase. So when we were going through all of the phases of mitosis earlier, we drew out a chromosome. Oh, what's going on? Here we go. So our chromosome had this centromere here in the middle. And that centromere, what binds these cells and holds them together is a molecule called cohesin. So we've got cohesin here in the middle. So it's cohesive, right? It's holding our chromosome together. And then we're also going to talk about our cyclin B that was in this G2 phase. So what we have going on in this checkpoint is anaphase promoting complex. So it's going to promote going into anaphase. So another word for anaphase promoting complex is E3 ubiquitin ligase. So if you remember, our E3 ubiquitin ligase was an enzyme that targets proteins with ubiquitin and targets proteins for degradation with a proteasome. So it's all coming together now. Proteasomes degrade proteins that we no longer need. So we're going to target the cohesin. Which was here. And we're also going to target this cyclin B because we don't need it anymore because we're done with this checkpoint. We want to move into anaphase. So we're targeting cohesin and cyclin B for degradation. Think proteasome. Remember, it's a little tube where the protein goes in one end and then it gets chopped up and spit out the other end. Make sure you can see this, sorry. Okay, so when that happens, we get rid of cyclin B, and then also our chromosome is able to split apart into two pieces, and then in anaphase, it's able to be pulled apart to the opposite sides of the cell. Okay, sorry that was a really long video, but we got through a lot, and I think that's it. Good luck studying for your finals, and let me know if you have any questions.